Hi, everyone. You're listening to Bug Talk, a podcast created by members of the entomology department at Michigan State University. In today's podcast, we will be talking to Inga Zasada. Hi, Inga.
Hi.
And I forgot to introduce myself. My name is Sophia. And it's a real pleasure to talk to you, Inga, because we have known each other for a very, very long time. We were just reminiscing that I think, first met in 2005. And we can get into that in a little bit. But before we talk about that time, I'd like to go back a little bit further in time and ask you to talk about your background, like where you grew up and what got you interested in biology.
Great. Yeah, definitely. And thank you for the opportunity to talk about myself and share some of my experiences with your listeners. So I was born and raised in Fairbanks, Alaska. Not a hotbed of agriculture, certainly, but a very unique place to grow up. When I was a kid, I was very much involved in 4-H. I did everything possible that you could do in 4-H. And that went from raising livestock to having a garden, learning how to cook and sew and all those kind of things. Just a really informative, important part of my life. And, that combined with having two parents who were both in science.
What kind of science?
My mom's a pharmacist and my dad was a scientist for the Forest Service. So my family actually has had 70 years of continuous service with USDA because my grandfather was a forester as well.
Wow.
So science was really just kind of part of my upbringing. I had great role models who showed me that I could be a scientist. I experienced kind of the nice lifestyle of being a scientist, not having to punch a clock and being able to pursue your own ideas. Mm-hmm. And I think that really resonated with me.
That must have been amazing to be a forester in Alaska. Oh my gosh, that must be a dream job for a lot of people.
That was a dream job for him. And we're going to talk about federal service a little bit later. But, one of the unfortunate things that happened to my father was the lab up there closed, because we are at the whim of congressional appropriations. So,
Sure.
That's a reality, or that used to be more of a reality, I believe, for Forest Service federal scientists, is moving around a lot, kind of following where there were positions. So that was kind of part of my upbringing as well. And actually, when I was a junior in high school, we moved to Oregon because my dad kind of saw the writing on the wall, and he moved down here to the Forest Science Lab with the family. And I ended up graduating and being part of FFA. Actually, that's when I got involved in Future Farmers of America here in Oregon. I was part of that. So again, just kind of really interested in agriculture. So I graduated from high school here in Corvallis, Oregon. And I really do feel Corvallis, Oregon has always kind of been my home. And that's why I was fortunate enough to come back here, but we can talk about that later. Did my undergrad at Oregon State University. I started in animal science, but I really decided that wasn't my group of people. And so I switched over to crop science, kind of enjoyed more working with plants. So I did that. I think another really informative part of my science background is I did a year exchange in New Zealand at Lincoln University. And that really got me interested in integrated pest management.
Was that as an undergraduate?
That was as an undergraduate. So my junior year I spent in New Zealand, and, IPM back then, this was the early 1990s, New Zealand has to import everything, right? So, everything they can do to create sustainable pest management systems that don't rely on inputs that come from out of country is of super value to them. So they were kind of ahead of us, certainly, I think at that time, in IPM systems. So I really got interested in that.
What did you work on there?
I mostly just took classes. I do remember though, and I think this is important for students, is, I went on some of these amazing field courses, where we went around the country and we looked at different production systems. We talked to different farmers, again, just kind of a tangible thing in my background that just really piqued my interest and kept me engaged in agriculture was those hands-on experiences.
So you didn't end up getting into research or any kind of labs over there?
Yeah, not at that time. My first, so my first lab experience was at Oregon State when I returned from New Zealand. I actually worked in a lab processing forage samples for protein analysis. So essentially I ground up alfalfa samples and ran them on a combustion, and I was all by myself in the lab, and I did this for hours and hours, which
That must have got you hooked. Oh my
I
gosh.
guess it did. The tediousness of science, right? I mean, there
Yes.
are some very tedious aspects to
Yeah.
what we do, and I'm fine with that. Being able to sit down and do a repetitive activity over and over. Does it? It's part of being in science, actually.
Yeah. You have to be good with that.
Yeah. So, I didn't have a deliberate path to where I am now. I'm kind of the type of person that kind of was presented opportunities and took advantage of them. I certainly knew when I graduated from Oregon State that I wanted to pursue a graduate degree. So I just started applying places, and I ended up getting accepted at North Carolina State University. I actually also got accepted at Montana State, but at that time I was like, I'll go to the East Coast and experience a different culture and, very different production systems. Back then they grew a lot of tobacco, which was of interest to me. So I decided to go to NC State, and I got my degree there in crop science and actually worked with the IPM specialist for the state, so an entomologist. But my project was focused on weed science and trying to reduce chemical inputs in weed control, specifically through no-till type production systems. So I worked in corn and soybean, and that was really my first experience to, field research, and designing experiments and collecting data and analyzing data. So that was a great experience for me. And the other great experience at NC State was, again, a very practical, hands-on course. And this was actually through the entomology department, where we had a group of students who, got into a van every Friday, and we drove around and we learned about production systems. We met farmers. We saw problems in real time. And again, this is something that's always resonated with me and has continued to keep me, I think, engaged in agriculture.
That's awesome. So I was going to ask, was this a master's program or did you go straight into a PhD?
No, this was a master's program, and I finished, and I was offered to go into a PhD, but at that time I didn't really know what I wanted to do, but I knew the other thing I always wanted to do in my life was be a Peace Corps volunteer. So I decided that was my opportunity. So I actually joined the Peace Corps about 6 months after I finished my master's degree, and I actually spent 2 years on the Maltese Islands.
I've
doing nematology. So that's where I learned about nematodes. My assignment was to be the nematologist for the island of Malta.
And they hired you without any nematology experience.
They did. I guess just having a master's was good enough. And I actually think that's probably a valuable thing to talk about, is that, you'll get a master's in entomology, nematology, plant science, but you can pivot. You as long as you have the fundamental concepts within science, don't be afraid to switch to a different organismal system. Or apply for a job that might be a little bit outside of your wheelhouse. Because I think if you demonstrate that you can do science and you're a critical thinker and you're motivated, I think people will hire you, even if you're an entomologist applying for a nematology degree or a nematology position. So,
I mean, not to denigrate you, but do you think they took you because they didn't have another nematology applicant?
100%.
Okay. So sometimes I guess the lesson is it's worth going for jobs that you're not qualified for just because they might not have another candidate that's qualified.
Exactly. Yeah. No,
I'm fields that are super small, like nematology, right?
Yeah, exactly. And, I think I probably got the position because I had a master's degree. I think they wanted someone, who was a little more mature, maybe a little more independent, to go in and try to figure out nematodes on the island and address that issue that they had. So.
And what was that issue? Or, I mean, they don't have much of agriculture over there. right?
No, yeah, most of
Like
their
a rocky
agriculture.
island with a few cities scattered.
Yeah, well, exactly. Well, actually, Malta is the fourth or fifth most densely populated place on the
Yep.
face of the planet. So there isn't a lot of land to grow food, but they do a good job of growing fresh produce for their domestic markets.
Mhm.
But when I was there, they filled a niche in Europe for early season potatoes.
Mhm.
So people in Europe would talk about Maltese potatoes, being available in the Netherlands or in Germany at a time of year when they didn't have domestic production of potato. And as part of that, there's always this concern about a quarantine pest, the potato cyst nematode, being distributed around the world. I mean, it's widespread in Europe already, and probably back then it was as well, but it's still considered a quarantine pest, and it's really something that you don't want. So some of the concern was that Malta was sending potato cyst nematodes with some of those nematode or those potato shipments to Europe, and it's a real thing. They use insects and diseases and nematodes to prohibit movement of produce. And a lot of times it might be political, but it still has to be addressed, right? And so that was kind of the main reason, was to kind of get an idea of how widespread potato cyst nematode was on the Maltese Islands.
And so did you like prepare ahead of time before going there, or you just went there and they told you this is the thing that you're going to work on, get
Right.
learning?
Exactly. Well, I did two things when they offered me the position. My mother and I first looked up where Malta was, because neither of us knew. And secondly, we would look up nematodes, because you don't learn about nematodes. You don't learn about nematodes as an undergraduate in biology, in spite of the fact it's one of the most diverse phylum, in the animal kingdom, but you just don't learn about them. And so I had to look up what a nematode was. And, beyond that, I really didn't prepare myself. I just kind of went over there. But what I did do was, when I got there, I reached out to nematologists in Europe and I asked for help. And the reason I'm a nematologist today is because I had folks in Italy and the Netherlands respond, and this is back when you wrote letters, respond that if I could get myself to Italy or to Wageningen University, they would train me. And so all I had to do was buy a plane ticket, and I showed up, and I spent 3 weeks in Italy and a couple of weeks in the Netherlands, learning nematology. So,
and that was enough.
Yeah, it was, it was intense. It was just, 8 hours a day on a microscope, either in a class in the Netherlands. They have a really excellent 2-week nematode identification course, or in Italy, I just sat side by side with scientists, and we just sat there and looked at nematodes all day. And yeah, it was enough. I went back and I spent a lot of hours by myself, looking at nematodes, learning how to draw. That's back when you had the drawing tube on the microscope.
Oh,
Remember
yeah,
those?
yeah.
Yeah. So spent a lot of time doing that. And I had the opportunity to do some extension work there. I had an opportunity to teach parasitology at the University of Malta. These were all things that I didn't think would be possible with a master's degree, to be able to teach at the University of Malta. It was just a really unique experience.
Did you enjoy living there? I mean, it's quite a switch going from Alaska and Oregon to Malta.
Yeah.
Like, you can't imagine much different.
Right. Yeah. And people talk about this who live on islands. There's island mentality, right? You live in these small islands, and it's, to be quite truthful, it's hard to integrate into these smaller communities that are, that have, that are on, I mean, Malta's an island that's 10 miles wide, 20 miles long. It's not a big place. And when you look like I look, with blonde hair, you don't really fit in. And so at times it was a struggle. It's certainly a developed country. It's not like I wanted for anything.
Yeah, and it's English speaking, so everybody speaks English, which is really handy.
Exactly. And I had water, I had electricity. I mean, I had a pretty cushy Peace Corps experience compared to what a lot of people experience. But on the outside, it looked like, being in a European country. But I think anytime you go to a different culture, it can be hard at times to integrate, to be accepted. Those are all things that kind of can be a struggle. So, and then living on such a small island, after 2 years, I pretty much, I think, walked every mile on that island when I was there. So,
Did you
yeah.
enjoy the sea?
Oh yeah, it was great. And there's this
Beautiful.
little island. Malta's made up of 3 islands. Well, 3 and a very 4th small island. But the island of Gozo,
Oh, so, yeah.
the smaller island up top, is really special. It's a beautiful place. It's very rural. Yeah, so you go over there, rent bikes, ride around. It was fun.
Have you been back?
I haven't.
Oh.
I would know I want to go back to New Zealand and Malta before I die. So
Yeah, it must have
resolved.
changed a lot since then.
Yeah.
So, so you were like, I'm guessing you were looking forward to getting out after the 2 years. It sounds like you were ready to leave and move on, right?
Yeah, I was ready to leave. And again, I've never been very deliberate in my path in life, but I really enjoy nematodes. I mean, I was hooked. I was hooked by
the 2 years like solidified in you that you wanted to work with nematodes and that was going to be your career.
Yep, that was it. I think between the kindness that I experienced from fellow nematologists and truly the small group of people who really care about each other. And I love looking at nematodes. They're super diverse, super interesting to look at. Yeah, I was hooked. So I again just started that, and finally at that point there was email. So I was able to get on email, and I emailed a few different folks, and ended up getting accepted at UC Davis in Davis, California for a PhD program in plant pathology. And again, a very important part of my life is my PhD mentor was Howard Ferris, a distinguished professor at UC Davis now. He was incredible to work with. He's sometimes, again, you have a certain relationship with your advisor and it's not all wonderful, but I look back and I truly couldn't have asked for a better mentor. I ended up TAing nematology 4 years in a row. I mean, at the time, I probably didn't really like that. But in retrospect, I am a super well-rounded nematologist. And a lot of it has to do also from Howard, and, he didn't focus on a singular thing in nematology. He had a very broad focus.
Was he focused on particular crops or crop groups?
No, he kind of is like what I am now. He worked on problems that presented themselves. He started off doing a lot of kind of conceptual modeling of nematode distribution and damage early on in his career. He worked in perennial systems, developing rootstocks with resistance to nematodes with breeders. And I think probably at the end of his career, what he was best known for was his thoughts on nematode ecology and understanding nematode processes in soil. So, he kind of dabbled in this, dabbled in that. And, I truly appreciated that, because I do think I won't ever be remembered for one thing. I'll probably be remembered just that I did a lot of different things. So, yeah.
And what did you work on for your PhD?
Yeah, I worked on biofumigation. So I worked on utilizing brassica plant material to control nematodes, and it was specifically in cut flower production systems. There's a big cut flower production system on the coast of California. So I did all my work. One of my field sites was literally on a cliff right next to the Pacific Ocean. So you would just look out across this beautiful flower field and you'd see the ocean, and it was wonderful.
Such a difficult life for you.
I know, it was hard. The drive was hard. I had to drive through San Francisco to get there, and that's never easy. But yeah, no, I had a great experience, and I worked with a collaborative team of scientists. The program was supported by methyl bromide alternative money, and it was run by a weed scientist, Clyde Elmore, and I did the nematology component of it. And that was great, because it really exposed me to a collaborative team. We had a pathology group working on soil-borne pathogens. We had our group working on nematodes, and then the weed scientists. And we were all working in this system of how can we maximize the benefits of biofumigation in what production systems is it going to fit? You know, we found on the central coast of California, where there's a lot of cloudy days, maybe that's not such a great place to implement biofumigation by itself. Whereas if you're in the Central Valley, of course, you can achieve biofumigation and solarization very easily. So yeah, it was interesting and fun.
So after your PhD, did you have any postdocs?
I did not. I was super, maybe fortunate, maybe not, because you know, you can always learn something from someone else. You know, I'm sure I would have benefited from a postdoc, but I was fortunate that I got my forever job with USDA ARS right out of my PhD.
So I'm wondering, what was it that your, you know, family history with USDA and the government, was that what got you interested in working for the USDA, or you just like hated teaching, 4 years of TA can get to you, or like all of the above? Like, what was it?
It was the fact that there were almost no nematology jobs at that time.
Yeah.
So yeah, I mean, it's actually really interesting to, and this might apply to entomology and plant pathologies as well, but I don't think it's as stark as what you see in nematology. I pretty much got one of the last nematology permanent positions for about a 10-year period. You look at the demographics of members of the Society of Nematologists related to age, and my cohort, which would be kind of your late 40s to 50s, there are very few of us. And that's because there were not positions being hired in academia, and nor in the federal government. I think there probably were a handful of industry jobs, but there was just a time when people didn't want to invest in nematologists. And I was lucky. I managed to get that job with ARS. But again, they're not a lot of
Was
people.
that the only position that you applied for, and there were no others, or how?
Well, actually, I applied for another position in Stoneville, Mississippi with ARS. And I went down there and I interviewed, and yeah, that was something else. I couldn't see myself in Mississippi. So, so there were 2 ARS nematology positions, and there's a really great nematologist in that position now. And I actually applied for a postdoc position at Colorado State University to work on nematodes in Antarctica. So, which I could have taken that opportunity, but which would have completely changed my trajectory. But you know, I want to be an ag. I really have been interested in agriculture, and I love agriculture landscapes, and I love everything about agriculture. So I think that was probably part of, well, plus a permanent job is a good thing, right?
Yeah. So, I mean, I'm just wondering, was it that they wanted to find out if there are nematodes in Antarctica, or they already knew and they wanted to explore more about them? Like, do we know?
Yeah, we do know there are nematodes in Antarctica. There's a dry valley. It's called I think it's what is it called? I can't remember off the top of my head, but there's a dry valley in Antarctica that doesn't get a lot of snow, and they actually study it because it's very small food chain. Essentially, you have a bacterial feeding nematode and then one other type of nematode, and so they look at ecosystem processes in this very simplistic, for lack of a better word, soil food web. So, and they've been doing that for decades.
That sounds like another one out there. I'm surprised you, after hearing your trajectory, I'm surprised you didn't go for that one.
I know, I know. I am, now that you mention it, so am I. I wouldn't be sitting here today if I would have.
True.
Yeah.
Yeah.
So
so you interviewed for 3 positions, it sounds like. Did you get all 3, or?
I did. Okay. Yeah.
So how was that decision then? I mean, it sounds like the Mississippi was out altogether.
Mississippi was out. Yeah. I remember standing in Mississippi going, why am I here? Anyway, again, it came back to my PhD advisor, Howard. You know, we sat down, we talked about it. And yeah, he really encouraged me to go to ARS and, you know, stay in ag research. So yeah, he was part of that process for sure for me. Yeah.
And how was it moving to the East Coast? I mean, I mean, you've been in North Carolina. It's not that
North
dissimilar, I guess.
Yeah, it was good. I am super glad I started my career there in some ways. ARS is it's a great organization to work for. There's no doubt. I mean, to have a job that's, 100% research, you go in every day, you self-motivate, you pursue ideas that are important to you, that you think are going to fill, answer important questions in science. But there's a lot of rules with ARS. And there's also, back when I was hired as well, ARS, 40% of the budget when I was hired with ARS in 2003 was congressional add-ons. So, a large part of the ARS budget was uncertain. So every year, president puts his or her budget up, and all those add-ons are taken out, right? And then they go to Congress, and representatives of Congress add all their special interests back into a budget. My position was created as an add-on. So my position in Beltsville was a brand new position to evaluate a biosolid amendment for nematode control. So essentially, it was an alkaline stabilized sewage product. And it was created through a congressional earmark from a congressperson from Ohio. And every year somebody would walk in my office and say, your position's not in the president's budget, so it created a level of uncertainty that could be uncomfortable.
Yeah, I mean, but it's not very typical that you're a USDA employee and your job is year to year. I mean, most people, this is their forever job and there's no such insecurity. So let's just put that out there, that this is a highly unusual situation for a USDA employee, right?
Yeah, exactly. And I should say that, once I was through my probationary period with USDA, which is a 3-year period, it affords you a certain amount of stability. So if that earmark would have gone away and had never been put back into the appropriations budget, they would have moved me somewhere else. So you're correct. I might not have been working on biosolids and nematodes anymore, but I probably would have been switched to work on something else.
Yeah, so that's where we met, at the USD in Beltsville, Maryland. I was a postdoc there, and you had been there maybe a couple of years when I came. So you were like introduced to me as the new nematologist, and I didn't even realize until just now that your project was so narrowly focused that you were initially hired on. And then, so you ended up only staying there for 5 years, right? So can you talk a little bit about your thought process, or what happened to land you where you are now?
Yeah. I'm super fortunate to have spent 5 years in Beltsville. First of all, living in the nation's capital is super fun as a young person. Second of all, you get to learn a lot about government, and you also are availed of a lot of opportunities that you might not get somewhere else. Being very easy to invite you to NEFA panels, for example, because you just have to go down the road. I was also very fortunate because, again, this is another example of someone who mentored me. My research leader, Dave Chitwood, was really a huge advocate for me and super supportive. So all of those things, I wouldn't give back my time in Beltsville because it's made me who I am today. But I do like to be in a more rural setting, a smaller setting, closer to agriculture. I think one thing that Beltsville, and this is my personal opinion, might struggle with as far as, getting increases in funding or getting stakeholder buy-in, is that you're not super connected to agriculture there, or I never felt like I was, because it was just a long way away and you're in this very urban center. And that's not where I wanted to live forever. So I knew the nematologist here in Corvallis was going to eventually retire. I actually harassed him every time I saw him at meetings. I would be like, when are you retiring? Anyway, so I knew him, and he retired and the position came up here, and I applied, and he was really great to me as well. He had, I think he was an advocate for me for getting this position. And as I mentioned earlier, I kind of consider Oregon home more so than Alaska. So I was lucky. I got the job, and I was able to return to Corvallis, Oregon. I love being here because, again, it's close to agriculture. It's close to the stakeholders that value what I do. But also you can drive 5 miles outside of Corvallis and have access to hundreds of miles of hiking trails. You're 45 minutes from the beach, you're 45 minutes from the mountains. It's just a really wonderful place to be. Plus, we're located on the corner of the Oregon State campus, which was the other big difference between Beltsville and here, is that I am part of the Department of Botany and Plant Pathology at Oregon State University. So I can have graduate students and mentor graduate students, which to me is super important. I don't know if you felt this at Beltsville, but I felt like there were no seasons at Beltsville, right? When you're in academia, you have semesters or quarters, you have students turning over. It kind of breaks the year up. At Beltsville, it just seemed like one continuous timeline.
It's interesting to me that the University of Maryland was just down the road from the facility at Beltsville, and so the proximity was there to another university. Just like for many USDA facilities, they are located on a campus or really close, and they have these kinds of relationships, but for whatever reason it never evolved that way, at least not that I know of, with scientists from the USDA over there, which is unfortunate, like you said, because I know that a lot of USDA scientists perform functions as a major professor or a co-advisor or an adjunct professor, but there are many, many different roles that they can fulfill.
Yeah, and I don't know if that what was going on with Beltsville and University of Maryland or scientists, there were just too many of us. I don't know that they didn't, that was just, I don't know, but I agree with you. I think it's unfortunate because, having young people come to your program and see things in a new light. I mean, it's just so important for me to be able to maintain enthusiasm about what I do. I can't imagine not being able to have students, that I can work alongside and mentor and learn from them. So that's super important to me. And that is a big difference between what I experienced in Beltsville versus what I'm experiencing here.
Yeah. And I'm curious to know whether you considered applying for other faculty positions around that time. If this person wasn't ready to retire, would you have considered going into academia?
Yeah, I totally would have. I actually applied for a job at NC State and did not get it, a professor position. I was ready to get out of Beltsville, so I would have, if there would have been a nematology job somewhere, I would have definitely applied for it. I think I'm okay at teaching and communicating. And again, I think I appreciate, folks like you who teach, because I understand how much energy it takes. I can only imagine actually how much energy it takes to do a good job. But I also think it keeps you really relevant. I mean, if you're engaged, and, I wouldn't have minded doing that at all. Or I wouldn't have minded having more of a extension type position, kind of like Marisol has. I think there's a lot of, and again, I also recognize how much energy it takes to run an extension program. But yeah, those are both things I definitely would have embraced if there would have been a position available. So.
And do you feel like you are able to meet that tie that you wanted to have with agriculture, being in Corvallis, even though you don't have a formal extension appointment? Are you able to tie your research into the environment there?
Yeah, and yes, I have. And the way I do it is by working with extension specialists in the state. So I interestingly, I live in Oregon, but most of my research is in Washington State. Because that's where the problems are. But everything that I do has an extension person involved, so someone who's lives in that community, organizes field days, does all that kind of stuff. I go and participate. So yeah, I have been able to achieve that, and kind of selfishly, I don't have to do a lot of the heavy lifting. My extension collaborators, organize a lot of stuff, and I, just show up. So.
Uh-huh. And
Yeah.
do you have students currently, or
Yeah, I have a PhD
registered?
student who just started, from Costa Rica. We're going to be working on Hopsis nematode with Marisol's group. We just got a proposal funded. So we're going to take a deep dive into how Hopsis nematode impacts hop production in the United States. And then I have a master's student who's, unfortunate for her, she started right at the beginning of COVID. So she's, a year and a half in, and it's been hard for sure. And I feel sorry for students who started during COVID, because it's really tough, but she's developing a quantitative PCR assay to identify different root knot nematode species.
Oh, wow, that would be really useful.
Yeah. And we're almost there. She almost has, her primers and her probes and everything working. We're going to get it validated. So and I think that if we can get that transferred to diagnostic labs at some point, I think that would really be an important tool in nematode diagnostics.
Yeah, because right now the standard mode of operation is to look through the microscope, and you need to have somebody qualified to recognize or identify those nematodes, right?
Yep, definitely. And with the ones that we're developing a quantitative assay for, the most important root knot nematode of potato here in the Pacific Northwest is Meloidigine chitwoodi, the Columbia root knot nematode. And the reason it's so important is that it actually infects tubers and causes feeding of the nematode and laying eggs, causes blemishes when the potatoes are fried. So you get these little brown spots. So there's pretty much zero tolerance for this nematode. So I, in addition to not having to have someone on the microscope trying to differentiate this nematode from the northern root knot nematode, I actually think a quantitative PCR would do a better job of detecting low levels of this nematode in soils. So I think that's pretty valuable as well.
And do you have to extract the nematodes from the plants to be able to run this assay?
Or just from the soil? Yeah, we do an extraction. I don't think you're going to get around that. I just don't see how you take a half a gram of soil and, I don't know, get a really good read on what you have there. So
that would be awesome if you guys worked out something where you just like put a piece of soil or plant in the tube.
No, maybe someday, but not now. So
awesome.
Yeah.
All right. Well, I want to thank you for coming on to the podcast and talking to us about your life. And it's such an interesting trajectory that you had into nematology, and hopefully it will encourage others to try and get into that field, because there are so few nematologists. And we didn't even talk about this, but nematology programs usually get thrown in with either entomology or plant pathology or some other departments. So there aren't very robust nematology programs across the country. Where would you say like the best nematology programs are across the country?
When you look at it like a group of nematologists, University of Florida has invested heavily in nematologists. I think there's probably 7 to 8 nematologists there, and I believe they're associated with the entomology department at University of Florida. The only strictly nematology department in the country is at University of California, Riverside. They still have a nematology department, and they have about
hanging on.
Yep, 6 nematologists there, a great group of people. So those are probably 2 that kind of have the most.
The largest.
Yeah, the largest. But again, before we got on the podcast, I do believe we're kind of in the second era of nematology. I think people are willing to invest in nematology research because we do not have a way to control these ubiquitous pests.
Yeah, I think it may have to do with the control methods that were taken away, that there's renewed interest in what do we do beyond methyl sodium and things like that.
Exactly. Yep. And 1,3-D and,
Yeah.
and I firmly believe nematologists need to be collaborating more with plant breeders because we have to figure this out on that end of things. And I just don't see enough of that, unfortunately. And we're just starting a big project here with a caneberry breeder using genome-wide association mapping to see if we can kind of get at those questions of finding regions of the raspberry genome that might be informative to be able to breed for, quantitative resistance to root lesion nematode. And I firmly believe, we got to get in that space because I don't see any other way of really combating nematodes if you don't have plant resistance. Yeah.
So hopefully we don't have that nematode, the potato one that you mentioned earlier.
No, you don't.
Hopefully it's not going to come here anytime soon, or
no, we do not
going to
survive.
survive.
Do not want it in Michigan. No.
Because we are big in shipping potatoes over here, so it would be a really bad one.
Yeah, I'm sure people are doing everything they can to make sure that nematode never arrives in Michigan. So
do you think it would survive in Michigan, or is it?
Yeah.
Oh, okay.
Yeah. So it's in New York and Idaho.
My gosh,
So,
it's closing in.
Yeah. So everything you can do for, only using clean seed.
Okay.
Yeah.
Good to know. Hopefully they're aware of this already. All right. Well, thanks again, Inga, for coming on. And I should also mention that you gave the department seminar a couple of weeks ago here, and thanks for doing that. And hopefully many of the listeners here were able to attend that and hear more about your research. But I will leave a link to your, I think you have like a USDA website.
Yep.
That has some of your publications up there. I will leave that in the show notes for people to check that out.
Great. Well, thank you so much for chatting.
Thank you. And thanks everyone for listening to the podcast, and we'll see you in the next one. Bye, everyone.
Bye.